My name is Tanaya Foster, and here's my neuron story. A neuron are the functional nerve cells in the nervous system. Inside the neuron is the cytoplasm, which is these green spots right here that you see. The green colored circles is the cytoplasm. And the cytoplasm is the clear inner fluid of the neurons in other cells. So there might be cytoplasm in other cells as stated. We understand that an event happens in cell A that causes a neurotransmitter molecule to be released into the space between cell A and cell B. So here's the space. The space between cells A, B, and C is identified as the synapse. Cells A, B, and C are connected by the synapse and dendrites, which are the bushy fibers that branch off of the cell. There are 4 things that can happen to cell A. The molecule can float away. That's also known as dissipate. The molecule can be taken back, and that's called reuptake. The molecule can dissolve or disintegrate by enzymes located in the interstitial fluid that will bind to the binding site, which is the receptor, which is on the receptor. The receptor is located on the dendrite of cell B. This is actually what we're most interested in when it comes down to the 4 things that can happen to cell A. The binding on the receptor. The presynaptic membrane, which is this green omega, is the section where it's right across from the synaptic cleft, and the neurotransmitter molecules are released into the synapse. The postsynaptic membrane, which is the yellow part, contains the postsynaptic receptors, which are the red little circle triangles that you see is the receptors. And the receptors, the neurotransmitters attach themselves to the postsynaptic membrane. So as you can see, here's the receptors falling in. Here, we're actually looking, I know the picture is not that great, but we're actually looking at the actual binding site, which is located on the ionotropic receptor, which is this. Here you see the ionotropic receptor. The ionotropic receptor is associated with the ion channels when activated. They send signals in the neuron by opening and closing the ion channel. Upon binding to the ionotropic receptor, the neurotransmitter molecules causes a pore to open that is permeable to sodium. So again, here's the ionotropic receptors. It's the red part right here. And then the ion channels is the yellow part that you see. But to furthermore even understand, we need to take a commercial break and understand what the difference between cytoplasm and the interstitial fluid is. So to actually understand the difference between cytoplasm and interstitial fluid is that the concentration of the ions inside and out the cell is different. In this picture, we have a large amount of potassium inside. We have a large amount of potassium inside than outside. The ions in the solution that travel down the concentration gradient is known as osmosis. When 2 charges are opposite, they will attract each other. The same charges will repel. This is known as electrostatic pressure. Eventually, the ions will move outside of the cell, as you can see. Outside of the concentration, gradient, causing less potassium to be outside and more sodium inside. So as you can see, it switched. So now this is greater. So here we're actually looking at the resting membrane The resting membrane potential will always be at negative 70. Looking at this picture, we can start to discuss the EPSP, the excitatory postsynaptic. When the pores are closed, and it can either depolarize or hyperpolarization will occur. Knowing this summation is when all kinds of neurons have been released. Explaining the excitation, you understand the summation. Here's the resting membrane. And here we can see the depolarization reduces to the polarization to 0. Remember, summation is when all kinds of neurons have been released. The ones regulating the sodium and potassium are voltage-gated channels. So when we start talking about thresholds of excitation, summation beyond the threshold produces a massive depolarization of the membrane. When reached, the membrane opens its sodium channels and permits sodium to flow into the cells. So the summation is when the neurons have stopped being released. So as you can see, there was a change here, and that's what we're talking about. Here's the end of the commercial. And moving on, we can start talking about action potential. And here's the transmission of action potential. So as we can see, the stimulus starts going in this direction. And we have some coming up and coming in, and they go up and go in, they go up and in. Here's the direction again, here they go up and in. The illustration is not the best, but you can see. And here we have, so here's a cell again, as stated in the beginning. Here's the cytoplasm. Here, the red circle parts is identified as the myelin sheath, and it's used to increase the speed of the axons. It's also used as an insulating material composed of fats and proteins. So as stated before, when we're going back to the